Good evening, Ajay.
Good evening, sir. You can start now, sir.
Yes, ma'am. Yes,
Good
ma'am.
evening, sir.
Good evening, Gana. Good evening. Okay. Good evening, my dear students. So you are all are warm. Welcome to the new session, a new chapter of the biological classes. The lesson is biotechnology and principle and process. Okay. So before that, we must know what is meant by, what are the basic terms comes under this heading, biotechnology. So biotechnology is the worldwide and also the advanced method that were used, okay, for plant as well as animals. Okay, here you must know what are the basic terms is that. Already you know very about each and every organisms are made up of a cell. Each and every cell, it contains accessory organs. What is maybe accessory organ? There is a center, there is a nucleus is there. Surrounding the nucleus, there is a cytoplasm is there. Cytoplasm covered with a cell wall or cell membrane, depends upon their plant or animals, isn't it? Inside the cell, there are so many accessory organs: the mitochondria, ribosomes, Golgi apparatus, endoplasmic reticulums, and also ectosomes. Sometimes it may be plant, as take an example, say chlorophyll molecules. Other components are also present in it, isn't it? So we must know. So what is by biotechnology? What are the technical terms that they were used? Okay. So already you know about the genetic material, DNA molecules, how it will be classified according to the chemical structures. What are the process which occurs? Okay. So that things we are going to discuss in this chapter very deeply. So this is also very important, the NEET point of view. So please watch carefully. Okay. So first one, basic terms you should know. What is maybe annealing? Already we studied these things also. What is maybe annealing? Annealing is nothing but the temperature related process. Otherwise called as when the double stranded DNA molecules undergoes the heat process. What happened? You know, the double stranded molecule in between there is a nitrogen basis is there. The nitrogen basis undergoes the heating process. What happened? You know, the bond is broken due to heat. So after that few minutes, what will happen? You know, it will cool under the particular temperature. So what happened? You know, again it will be united and form as a double stranded DNA molecules. That condition is called as, what is it? Annealing. See that one? The annealing is the process of heating and the slowly cooling of the double stranded DNA to allow the formation of hybrid DNA molecules. You know a little bit what is meant by hybrids? Hybrids nothing but the two different genes united. Okay, united and form as a single gene. That is called as, what is that? Hybrid, isn't it? That things we may contact, we may produce with the help of the annealing process. Okay. Next one is bacteriophage. Already you know a little bit. What is meant by phage? Phage is nothing but the virus molecules. Bacteria means it's nothing but the prokaryote. It is a lower organism. Take an example, prokaryote example, bacteria. So, okay. A virus that infect, otherwise called as the bacteriophage, means the phage that infect the bacteria is called as bacteriophage. Okay. So there are so many bacteriophages have been reported here. Lambda phage, okay, and also TMB mosaic virus is also, it's one of the polyhexohedral, isohedral head. That things also comes under the heading is known as bacteriophage. Okay, next one is capsid. You know about the what is meant by capsid? Already you studied in the TMB, tobacco mosaic virus or cauliflower mosaic virus. It is made up of two major components. Is there? The two major components, main one is nucleic acid. The nucleic acid is whether it may be DNA or RNA molecule, depends upon the plant or animals. Suppose if it is plant, okay, it contains only the RNA as a genetic material. Suppose if it is animal, okay, so DNA as a genetic material. In this point of view, it may be differ in some of the cases. Taken examples, COVID virus is an animal virus. At the same time, HIV also a animal virus, isn't it? Both nucleic acid, both the viruses, they are having the nucleic acid is RNA as a nucleic acid, contain the 2 copies of single stranded RNA molecule with the help of reverse transcriptase enzymes. Already we studied that things also. Okay. Here, why we are studying that? Another one is called as nucleic acid is there, and the another part is there. What is that? You know, the capsid is nothing but the protein coat. The DNA molecules covered with the protein coat is called as what is that? Capsid. Okay. Suppose take an examples of TMB mosaic virus. It is covered by a capsid protein molecule, and also it is made up of more than number of capsomeres. Okay. So these are the 2 things is very, very important. So once again, repeat for a capsid is the external protein coat of the virus particle is called as capsid. Take an example, HIV also, there is a outermost protein molecule is there. It is nothing but the spikes or hooks. Okay. The 2 types of protein molecules is that. One is called as a glycoprotein, GP. Okay. And 21, and another one is called as a 141, like that, you know. Different types of capsids also present. And the next one is called as cloning. What is the meaning of cloning? You know very well about the iron wilmet, isn't it? One of the scientists. So what they did, you know, the research work, during the research was what he obtained or observed, you know, one of the goat, he has chosen a goat, and also finally he get the 2 dolly goat during this process. That means the cloning is nothing but the single parent only. They will not fuse the male as well as the female gamete together. Okay. Only one parent is enough for the cloning process. They isolate the cell and also remove the nucleus from the cell, and also they will introduce the new one, what they are going to develop. The desirable characters should be introduced inside the cell and replaced in the same organisms. Okay, that is called as cloning. See here, the cloning, it is a process of producing many identical copies of organism and cell. Take an example of tissue culture is also one of the cloning method, because a single cell, it produces a number of platelets, 1,000 millions of platelets in a single test tubes. That condition is called as totipotency. Already you know a little bit. What is meant by totipotency? A single cell, it will produce the entire plant is called as that capability, that ability, the potentiality is called as what is that? Totipotency. You got it? Okay. So
Yes.
go to the next. Okay. So go to the next one is called as media. So what is my cloning vector? Already you know the vector is nothing but the additionally support for the cloning process. Okay, already we studied what is my cloning vector. It means the plastid is also one of the cloning vector. The factor phages and cosmids. These are the examples of the cloning vector. Suppose if we want to introduce, means we are selecting the DNA molecule from the different organism. Take an examples of A or B. Okay, already we saw it in the yesterday classes. Restriction endonucleus, we made a cut, the particular polyandromic sites, and remove it. A desired qualification, desired quality of the character will be introduced. Both they need which one, you know, the molecular scissor, that is called as a restriction endonucleases for cutting process. That's why it is called as molecular scissor. The desired quality is also introduced and also united with the help of the enzyme is called as ligases, DNA ligases. Okay. After that, what they will do, no, they introduce into the host. Any one of the host is essential. That host is called as cloning vector. After cloning process done, you have to introduce into the further process with the help of the vector. So what are the vector used for the cloning process? Plasmids, bacteriophage, and cosmetics is an example. See that one? The cloning vector, a small self-replicating DNA molecule into which foreign DNA. So both are. This is a DNA molecule with the foreign DNA. So the sample 1 and sample 2 is inserted in the process of cloning genes or other DNA sequences. So not only that, whatever it may be, what are the desired quality, what are the desired characteristics we can, we need that can be laboratory we may do or we made it. Okay, so this is called as a plasmid. This is a structure of plasmid. Okay, it contains the characteristics of so many things is there. Here, what are the instruction given here? The antibody resistant genes. Take an example, this red color part, antibody resistant gene is there. Suppose if we want to isolate this part only, because we need tetracycline, ampicillin, and so many penicillin. Okay, antibody resistant gene also present in the plasmids, isn't it? We have to isolate the exact antibody resistant genes from the plasmids. So that time what we will do in laboratory aspect also, they are using the molecular scissors. The particular part will be completely removed, and also it is joined with the foreign DNA. After that, they introduce into the which one host. Okay, so this is the example. Next one is called as complementary DNA. It is simply called as cDNA. Already we studied yesterday also, isn't it, my dear? A DNA strand formed from the mRNA. This is very, very important. Complementary DNA means, what is the name? The exact molecules of the DNA molecules strand formed or produced from the mRNA by using the enzyme reverse transcriptase. Okay. So the reverse transcriptase is also called as, what is a complementary DNA, otherwise called as small c DNA. Note it down, my dear student. With the help of the enzyme, which one reverse transcriptase. Reverse transcriptase means, what is that see here, RNA, okay, to DNA. So this process is called as, what is that complementary DNA. With the help of the enzyme, reverse transcriptase. Downstream processing, another process is also very, very important. Separating and purification of the product from the fermentation process. Downstreaming process also is present in the DNA replication process. Already we studied that things also. Okay. And next one is endonucleases. What is the meaning of endonucleases? If you want to cleave the molecule, suppose take an example, 2 strand of DNA molecule present in it, that 2 strand, it will be cleave internally, means we were used one of the enzyme is called as, what is that? Endonucleases. What is the main action of the endonucleases? An enzyme that catalyzes the cleavage of DNA at internal position, cutting DNA at a specific site. So here, as I would like to say, this is an example. Okay, see that one. So this is a DNA molecule. Okay, this is a DNA molecule. Take an example. So this is considered a DNA molecule. There is a nitrogen base is there. So these are the nitrogen base. Okay, so these are the nitrogen bases. Opposite is also there. Opposite is also, there is a complementary basis there. Already know very well about that things. Here starting from the 5-N, this one is starting from the 3-N. So here 5-N, here the 3-N. So the endonuclease enzyme, what they will do, the endonuclease. See that restriction endonuclease. Okay, then restriction endonuclease. What they will do, they identify the particular place of the polyandromic place. They made a cut here. Here they made a cut. Okay, as a result, the DNA molecule, how you will obtain, see the structures. The structure will be like this. Already I saw it. So the structure will be like this. Those two stand, you will obtain like this. Okay, it will be, the cut is made here only. So this position here, and this is the position now here. Okay, so the cut is made with the help of the enzyme endonucleases. After that, in this portion, so we can remove this portion. Okay, we can remove it, this portion. See that one? We can remove this portion. Okay, and completely remove it. And we may add the plasmids, another gene. This is one gene. This is sample A, consider as a sample A gene. This one is called as foreign gene, F gene, otherwise called as sample B, like that you can. It made a cut. Okay, you made a cut in this position. For example, see that one? You made a cut in this position. Okay, the particular portion, restriction for the enzyme. This is considered as a restriction genes. This is replaced here. Okay, for the process. Okay, see that one? So as a result, what happened? This is completely. So erased. So this gene is, what is that? This is the part B gene is also introduced in this area. So this gene is A, this gene is B gene. So both are united together. This condition called as, what is that? Cloning. Okay, so that process is called as cloning process. After cloning, what happened? They both are united. For now, with the help of the enzyme is called as ligase. The both are united. How it will be? It will be like this. Okay, it will be united like this. With the help of the enzyme is called as, what is a ligase? Ligase is a enzyme. Okay, that is called as DNA ligase. Simply we may call as, what is that? DNA ligases. DNA ligase. So after the ligation completed, that means the foreign DNA of the both A and B's DNA molecule united together, it introduced into the E. coli. We need host, then only the replication process also possible. Then only the replication process also. After this formation, we have to introduce the E. coli bacteria or bacteriophage from here to here. Okay, the structure will be how it will be. Same things is that you have to split into two categories like this. See that? Okay, the structure will be, so here, this one, also another one is like this, isn't it? Now both the gene present inside the host, that is called as maximum bacteriophage or bacterio, otherwise called as a E. coli maximum. They were used as a vector. Okay, so here what happened, both the genes are amplified inside, otherwise called as replication takes place. Okay, replication takes place within the host. Once again, repeat for you, my dear student. Endonuclease is one of the enzyme it used for the rest cutting molecular scissor, act as a molecular scissor. This is the DNA molecule. It cut here the polyandromic genes. After that, the A gene is cut and B gene is also cut with the help of the molecular scissor. Both are united. And after the united, you will get the structure like look like this one. Again, it enter into the coast. Again, it enter into the coast. So the replication process also done by the coast. What happened? You will obtain the both the gene in a single organism. This is the process. Do you understand my idea? Okay. So.
Yes, sir. Understood, sir.
Okay, dear. See that. So this is called as endonucleases. Exonucleases. It is also another type of enzyme. Okay. That is endonucleases means inside. Exonuclease means it's outside like that. An enzyme that removes the nucleotide from the end of the double stranded DNA molecule. If you want to eliminate or remove a particular nucleotide with the help of the enzyme is called as exonuclease. Okay, so this is very, very important, my dear. So don't forget. The complementary gene and downstream process also there, and endonucleases and exonucleases. These are the important technical terms that they were used in these lessons. Okay, see to the next one. See that one. So this is the restriction enzymes. So this is the polyandronic gene. See here, GAA-TTC. Okay, see here in this part, GAA-TTC. When you are reading the left to right, you have to read the right to left. Both are similar in nature. Okay, the cut is mad like this. Already you saw it in the diagrammatical aspects. After that, it is removed and also addition of new one. Then introduced into the host. So this process was done by which one, the restriction endonucleases. That's why it is called as molecular scissors, particularly E. coli. Don't forget, my dear students, E. coli. All the living organisms, one of the most enzymes present in the all the living organism. Taken examples in the lower means bacteria to higher animals. Which one of the enzyme means E. coli? Don't forget. Ajay?
Yes, sir.
Okay. One of the most enzyme present in the all the living organisms means which one of the enzyme, my dear?
Sir, E. coli, sir.
E. coli. Likewise, I asked one more question is that. Okay. Which
Sir?
protein? It's
Sir?
also which protein molecule is also a universal? Which protein molecule is also called as a universal protein?
Sir, not sir, sir.
None. Okay. What about the universal enzyme now?
Sir, E. coli.
Right. So universal protein molecule, don't forget, my dear, please take a small notes or notes. Okay. So what is that, collagen? What is the meaning? Universal protein is called as collagen. COLLEGEN, collagen. Okay. The collagen is a mostly abundant protein molecules present in the all the living organism. All the organism, it contains the one of the protein molecules, a collagen. That's why it is called as universal protein molecule. Sometimes they ask the question, just go and check it in a 2016 or 17 or 18. These three questions. The universal protein collagen is made up of which one of the following chemical substances they asked? That is called as chondroitin sulfate. What is the meaning? See, my dear. So one of the universal, okay, universal, sorry. So I will text it, then only it will be better for you. Universal protein, okay. Which one is the universal protein, my dear? Collagen. Okay.
Collagen, sir.
Okay. So what about the chemical name of the collagen? Just to imagine, this is chondroitin sulfate. CHONDRIDIAN, chondroitin sulfate. SULFATE. Okay. The chemical name of the collagens is called as what is it? No, chondroitin sulfate. At the same time, universal, what is that? Universal enzyme. Okay. The universal enzyme is called as E. coli. So what is the meaning of E. coli? E. coli.
Sir, it's a universal enzyme, sir.
Yeah, you know that the expansion of the E. coli, that is very, very important. E. coli expansion. Escherichia coli restriction enzyme. What is that meaning?
Sir,
Ah,
it's a restriction enzyme, sir, but I don't know the full form, sir.
yeah, E. coli means Escherichia coli. E. coli.
Yes, sir.
Okay, restriction enzyme. Don't forget, my dear. Okay. Okay. See, this is the two things is very, very important. So many times they were asked the question. E. coli is nothing but Escherichia coli restriction enzymes. Okay, universal protein collagen. The collagen is made up of chondroitin sulfate. Okay, the chemical name of the collagen is called as chondroitin. Mostly they were used the connective tissues. Connective tissues means what is that? You know, there is a bone. Okay, just imagine. This is considered as a bone structure like this. Okay. See, the structure is look like this one. The bone is look like. Okay, so this is the bone. Just imagine. So this is also another knee bone. Take an example. In this part, okay, in between this there is a synovial fluid is there. The synovial, VI. Okay, here I will type. It's better for you. So synovial fluid. Where is the synovial fluid? In this portion, because then only it may be easily flexible. Okay. So what we are going to discuss in this part, the chondroitin sulfate is one of the connective tissues, means the bone as well as another bones also, what is that? Connected with the one of the specialized tissues called as what is that? Chondroitin sulfate. See that one? In this portion, it is covered with a small protein molecules, the both. Okay, so I have to differentiate, then only it will be better for your understanding. Okay, so in this portion, there is one of the connective tissues is bone as well as the here also. Okay, so here also there is a connected. This is called as what is that? You know, so universal protein that is called as collagen. Simply we might call as collagen. Okay, in between what happened? There is a fluid is that fluid is called as synovial fluids. Then only it can be easily just like a lubrication process. Okay, we can move from the upstairs. We can easily move. Okay, there is no. Suppose in old person, what happened? You know, the collagen completely degenerated, destroyed. Okay, so that's why they got pain, knee pain. Okay, due to this one. So degradation of the cells as well the fluid is also what will happen. You know, reduce the fluid amount also. Okay, so this is also denaturing of the collagen fiber. The protein molecules also denatured because one of the connective tissues with the knee. Not only here, so many places also. Okay, so this is very, very important. Okay, my dear. Understand? Ajay?
Yes, sir. Understood, sir.
Okay, okay, dear, my dear. Listen here. So next one is what is it? You know, see that one. A difference between endonucleus and exonucleus. So see that one. This is the endonucleus. There is a cut already told within the molecules only. Exonucleus is it. So this is a fragment of DNA polymerase 1. The fingers. Okay, these are the fingers forms and the exonucleus also. What is the main use of the exonucleus? This is also a removal of the particular DNA molecule from the segment with the help of the exonucleases. Okay, this is the structure. Endonucleus means what have you know? So we make a cut in this person with the help of the. This is also Hind 3. So another enzyme is also there are so many enzymes also involved. Hind 3 is the mostly used. Hind 3. This is also a very, very important question, my dear. Which restriction endonuclease is used for the DNA? as a molecular scissor. It acts as a molecular scissor. Hind3. Not only this one, E. coli is also there. Okay. The E. coli is a universal protein molecule, so universal enzyme molecule, because enzymes are made up of protein. Okay. This one is also. Exonuclease means what is it? Completely removed of the DNA molecule. Okay. From the fragments. Okay. See that one? You can understand. Basic differences. What are the basic differences? Means this the function is also different. Cleavage spot is also different. The end and the properties. What are the examples? Okay, these are the basic differences between the exonuclease and the endonucleases. First, they have told exonucleases. The exonuclease, what happened? Cleave the DNA sequences from 5' or 3' and one by one. Usually non-specific in function. According to their function, the exonuclease activity on the DNA molecule, how it will be? The cleavage DNA sequences from the 5' or 3', any one of the dash. Okay, any one of the side, whether it may be right side or left side. How it will be? And one by one, usually non-specific in function. But in this category, endonuclease act upon the phosphodiester bond. Phosphodiester bond means what is that? The cleavage occurs in this area means what is that? Here there is a nucleotide phosphate molecule is there. In between the nitrogen base is also that. So the phosphate molecules also cleave, and here the nitrogen base is in between these two molecules. So that the binding of this part, okay, also cleave. Then only you will get it the sequence. The DNA and RNA sequences target only specific. Okay, target means the polyandronic genes. So that is very good. Here, any one of the directions. Here, a specific site only it cleaves. That is the difference between exonuclease and endonuclease. Cleavage spot, cleavage free terminal nucleotide sequences. For example, so free terminal means just imagine. Okay, just imagine. So this is a DNA molecule. Here, the free terminal is there, isn't it? Suppose if it is considered here. So this one is called as 3', sorry. So this is considered here 3' means here 5', isn't it? 5'. Here 3' again. Here 5'. The cut may be any one of the corner only, free dash. Okay, this one is considered, so last one. Okay. So this one, it can be easily removed because it is a easily removable part. Any one of the part, 5', here also there is a 5', here 3', here also 3'. Okay, we can remove in this part. For example, adenine is here. Suppose here guanine is there. For example, the guanine means here cytosine is there. Adenine means here thymine is there. Okay, we can remove in this part or in this part because the edge only. Okay, we can remove it. But at the same time, which one you know? In the endonucleases, already we discussed the DNA molecule. It can cut a specific site only. In this portion, maximum, what is the polyandromic gene sequences only? It can be removed. Okay, so that is very, very important. Right? See that one? And then the endonucleases nicks at a specific position with the DNA that already we discussed. And the end, the cleavage always result in the formation of sticky end. So here there is a very, very important one. The exonucleases, what is sticky end? It can be easily fixed with the another part, sticky end. What about in this part? Cleavage result is in either sticky or blend end. Blend end means what is it? There is an extra end is there. See that one? So that things also you can easily understand. This is considered. So this one. So this one is called as what is that? Exonucleases or endonucleases, isn't it? This is a sticky end. It can bind the exact copy of the another DNA molecule from the another part B. Okay, foreign particle. It can be easily bind. But in this case, what is that? There is a blend end. Is that there is a blend end? So that is also reported here. Okay. So cleavage result is the sticky or blend end. The properties, see the lack defensive properties, again entry of pathogens. Lack defensive properties. Exonucleases, there is a lack of defensive properties. But in this category, have defense properties against the antigens. Here the pathogen, here antigens, both are same. Pathogen produces antigen, isn't it? When it enters any one of the foreign particle into the body, what will happen? Automatically it will produce antigens. Antigen is nothing but the invading bacteria. It produces a protein substances. That protein substances called as antigens. Okay, Ajay.
Yes, sir.
Okay. Then that antigen is act in your body. As a result, they emit the symptom. They create the symptom. They produce the symptoms. So then only you will fall into the sick. So after that, what happened? You are going to the doctor for the reference. Doctor provide antibiotics. So the antigen antibiotics united together. Suppose if it is correctly matched, what happened? The sick, it will be clear within 2 or 3 days. Suppose if it is not, means what happened? You have to take another part, you have to change the antibiotics. That is very, very important. Okay. So this is, and the last one is the example has given is exonucleases. They have given as a exonuclease 1. Endonucleases they have given as a E. coli or 1 E. coli. Okay, E. coli, Escherichia coli restriction 1. So restriction 1 means what is the meaning? Restriction 2, restriction 3, restriction 4 is also present. Don't forget, my dear. Exonuclease means, exonuclease 1 means, and also another subdivision also is there. Don't forget. Okay, the main one is that what is that? Endonuclease means first you should note it down. So E. coli 1 only. Exonuclease means they have given which one? See this one. So here hint 3 is also there. Here the E. coli 1. These two are restriction endonuclease only. So the hint 3 is also one of the restriction endonucleases. This one is exonuclease. Exonuclease, what they have given? DNA polymerase 1. What about here? They have given is a exonuclease 1. So that is denoted as a exonuclease polymerase 1 only. Okay, don't forget, my dear. Next one is a gel electrophoresis. Last class we studied this one. What is the meaning of gel electrophoresis? What is the meaning of gel electrophoresis, my dear? Gel electrophoresis is nothing but one of the instruments used for the separation of molecule. What is that? Separation of the molecules. Okay. See that it is a technique to isolate the DNA fragments with the help of the gel agarose. Gel agarose. With a nylon membrane, otherwise called as nitrocellulose paper, and also probe. These are 3 is very, very essential for the DNA fingerprinting. DNA fingerprinting. Don't forget, like it. Okay. So these are the important. Gel electrophoresis is nothing but the technique. Okay. See that the technique already we saw it, isn't it? This is the technique. Gel electrophoresis. So the gel electrophoresis means what does it mean? There is a plate, is there glass plate? The glass plate loaded the gel, agarose gel. From there is a buffer system is there. Buffer is also filled in this area. There is a sample place is also, that is called as pit. Otherwise called is a, what is that? Pit is also there. We have to load the sample with the A, B, C, D here. I think there are 6 pit is there. After the complete setup, what we have to do now? We are passing the electric charge on it. So negative molecules move on to the positive side. See that one, loading the molecule, that is called as loader. Okay, protein analysis, apply of current. So what is that? There may be a analysis. They have given as a, see that there is a band obtained from the, after that we may get it these structures. Okay, then the nitrocellulose already know about, you have to fix with the nitrocellulose and then ethiobromide. And finally, it may be entered into the, what is that? Radiographic methodology also already know about that things also already we discussed. Okay, this is the one. See, and the next one is called as ligases. Now only we studied. What is my ligase? Ligase is nothing but the joining. So ligase, an enzyme used by the genetic engineer to join and the cut ends of the 2 different double-stranded DNA. Cut the end, already we saw it, isn't it? So where there is a cut we made, that cut is also joined with the foreign DNA molecule with the help of the enzyme is called as DNA ligases. Ligases nothing but the linking of 2 genes. where there is a cut, where there is a, we made a cut. So they both are united. You will get the single standard. Again, it will be get the normal one. That is very, very important one. And third one is called as micro injections. This is very, very important, my dear students. Okay. The micro injection is nothing but what is it, you know? For example, take an example, see any one of the cell. This is consists a cell. Okay. See that one. Then only it will be better. See that, my dear. So clear. See, this is my dear. So this is consists a small bacteria cell structure. Okay, this is consists a bacteria. Inside what they are having, you know, so there is a extra chromosomal body. Don't forget, my dear, extra chromosomal body is there. The extra chromosomal body is called as what is it, you know, plasmid. What is that name? It's called as, this one is called as plasmid. This is consists a bacteria. Okay, this is bacteria, this is plasmids. Okay, here what we are going to study now, ligases. Okay, how it will be? See this one. Here we have to make the cut. So already you know about this structure already. We saw it in the previous sessions. Okay, so this is the plasmids. Okay, this is the plasmids. We have made the cut in this position, in this part. Okay, this part is completely removed. Excuse me. Just a minute, Ajay. Okay. Sound what is it? Jay, sorry. So here,
Yes, sir, I'm there, sir.
sorry, charge is reduced. That's why. Okay. Okay, listen here. So this is a. So here, what happened? This is the plasmid. So just imagine. So the extra chromosomal body is called as plasmid. Already you know about. We made a cut in this position. Okay, we made a cut with the help of the restriction endonuclease and remove it. So as a result, the structure will be look like this. Okay, so we'll get it. So the structure will be the like this. Okay, so this part is completely removed in this portion. So we are adding the another DNA. Already I told you the things also. So we are adding the another desired quality or desired characters. So this is also again enter into the which one, you know, for the replication process. Again, it introduced into the host. Okay, so it is a different host. So E. coli host is also act as a E. coli. Sometimes it may be a phage also, otherwise called as bacteriophage. The bacteriophage already you know how it will be look like a structure will be like this, isn't it? The bacteriophage, it has a 2, 3, or 4. How many structure is there? There is a 3 part is there. Okay. So here there is a DNA molecule is also there. DNA molecules. Inside there is a nucleic acid. Okay. In this part, what is that? You know, the protein code is there. This is the DNA molecule. This is the protein molecules. This is the DNA molecules. Okay. So DNA molecule. Okay. This one is called as DNA. And in this area, this is the collar. This position is called as collar, otherwise called as neck. This is called as what is the body part. This is the plate, base plate. Okay, here there is a hook-like structure is there. This is called as what is a hook-like structure. This is the entire structure of the bacteriophage. Okay, so these are it is also act as a host. Already told you. So E. coli is also one of the host. Otherwise called as bacteria is also act as a host. And then second one is what is that? You know, the phage is also act as a host. Okay, so these two things you must note it down, my dear. This is very, very important. Okay, here, so we studied that ligases. So ligases, what is that? The micro injection only. The micro injection, entry. What is the meaning of micro injection? Means a technique of the injection DNA or RNA into the nucleus of the protoplasm of the cell, which the help of the micropipette. Micropipette means what is it? It is also, if you want to introduce a minute quantity, millimicron and microliter of the DNA molecule into the plasmids. How it will be? See that one. So it will be better for your understanding, because it is very, very important, my dear. Okay. Take an example. So there is a cell is there, isn't it? Just take an example. This is considered a small cell. The center there is a what is a nucleus is there. Just imagine there is a nucleus. Okay. Inside what happened? No, the DNA molecule is also there. The DNA molecules. Just imagine this is the DNA molecule. Okay. We are going to isolate from this is a normal one, and another one is called as protoplast. Okay, this is considered as a protoplast. Protoplast. The protoplast is also, it has a characters of the particular genes. Okay, this is also very, very important. Okay, the protoplast. Just imagine the protoplast is also important one. Okay. Okay, this is the protoplast. Okay, here how it will be hybridization takes place. This is considered as a sample taken from the tobacco plant, any one of the plant sources. Take an example. This one is considered as a plant source. This one is plant. So another one same also taken from the. Okay, so this is also another cell. The DNA molecules also, otherwise called the protoplast, is also taken from the animal sources. This is bee sample. This is animal. Okay, animal. Okay. So we have to isolate these DNA molecules, this plasmid molecule outside. With the help of so many techniques they were used, cellulose. Okay, here the black is I have used. So I have to choose the black. And so we have to isolate this plasmid. So we have to cut this cell wall here also. So then only it comes out, isn't it? So after that, it will come out. Okay. Here we have to find out which characteristics we need, like that. For example, take an example. Jellyfish, do you know, my dear friend? Jellyfish. Ajay? Hello? Ajay? Okay. So here we have to make the cut in this portion, A. Okay. Here we made a cut in this portion, S. C. Both are removed. Okay. This is one part. Okay. And united. And another category, what is it? You know, the plasmid, we have to introduce the, so for example, take an example, any one of the animal sources, you have to introduce it with the help of the micro injection, otherwise called as micropipette. Micropipette. P-I-P-P-E-T. Micropipette or gun. What is that? Electric gun is also. Maximum they were used the electric gun. The micropipette, it is also, the micro injection is also, what happened? It is a small needle-like structure is the needle-like structure. What happened? No, it focus on the, what is that? This is the micro injection. Okay, this is the micro injection. Here loaded a component. What are the DNA molecule from the any one of the A or B? It can be targeted to the organisms. Any one of the desired organism, it may be loaded. Okay, it may be loaded. These are the molecules loaded inside. Targeted means what is happening? It may be targeted exactly copy this one. So as a result, what happened? No, the combination takes place. Inside what happened? No, the combination takes place. The recombination. Okay, recombination it takes place. See that one? So you'll get it. So the target cells also reach like this. So here the combination of both. they were used one of the technique, the micro injection or the micro pipette, okay, used for the loading of the target plates, target organs. Okay, so this is very, very important, my dear. This is also made up of gold. Okay, we will see you in the later. See that one. A technique of injection DNA or RNA into then nucleus or protoplasm of the cell with the help of the micropipette. Second and next one is called as origin of replication, simply called as ORI. Already we discussed that things. Origin of replication, where there is replication starting place, that gene is called as gene or ORI gene. See that one? It is a sequence from which replication start, and any piece of the DNA when linked to the sequence can be made to replicate within the host cell only. The replication starting place. This is very, very important. The ORI is nothing but the replication starting place is called as what is that? ORI. Okay. So this is also very, very important. Already you know about the replication starting places are also any band. Take an examples of 5' to 3' or 3' to 5'. Okay. So one is called as leading strand, another one is called as a lagging strand. That things also already we discussed in the replication process. Okay. Particularly semi-conservative method. How many minutes it will take? 38 or 30 minutes for the things. Okay, so that thing's also very, very important. So this is already we discussed. Polyhydronic sequences. This is very, very important, my dear. Okay, gel electrophoresis system already we saw it. And polyhydromic scenes already we discussed the complementary DNA sequences that are the same when you reach stand is read in the same directions. These sequences act as a recognizing site for the type 2 restriction endonucleases. Polyhydronic, don't forget me. The restriction 2. Okay, so they can find out the 5' to 3'. 3' to 5' restriction enzyme 1. 3' to 5'. Once again, 5' to 3' means what is that? The restriction enzyme 2 only act upon it. Okay, so this is a very, very important. Next one is a plasmid. Already told you the plasmid, nothing but one of the extra chromosomal self-replicating circular DNA molecule. This is very, very important. Extra chromosomal self-replicating circular double-strand DNA containing some non-essential genes is called as what is that? Plasmids. Okay, don't forget it's mostly found in the E. coli or some other lower bacteria also. Okay, and also the plant also it has the plasmids, but it has a extra chromosome. What is the meaning of chromosome? It containing genetic sequences of the particular organisms. Okay, so that is very, very important, my dear. Next one is a polymerase chain reaction. Already we studied in the DNA fingerprinting. Okay, the polymerase chain reaction is also one of the techniques used for the amplification of the desired genes in number of copies within the lab is called as polymerase chain reaction. Polymerase chain reactions. The action of the enzyme polymerase to produce the many copies. This is very, very important. Many copy of the DNA molecules produced at a high temperature. That is very, very important. Then only we'll get the polymerase chain reactions. Polymerase chain. Polymerase means more than one. With the help of the enzyme polymerase. Chain means it's also continuously doing. Okay. The reactions also continuously with the help of the prop. The primer is also, DNA primer is also very, very important. Then only the reaction will be continued. Otherwise, what happened on? It will not continue. And also some of the markers also that were used. Some of the markers, the DNA marker is also used for the prop. Then only we can identify which one is the newly formed one, which one is the old one. We cannot be able to differentiate. So that's why they were used the prop. See that one? Polymerase chain. So this is the step is called as polymerization reactions. So this is the DNA double standard structures. Here, what are the enzymes or what are the components essential for the polymerase chain reaction means, first of all, you should know DNA molecules, very, very important, double standard molecules. Addition with it, there is a primer is also very, very important. And also NEET is, okay. This is a triphosphate. And the polymerase enzyme is also very, very important. These are the three components essential for the polymerase chain reaction. First of all, what they are going to do now, denature. How it will be denatured in slow? It's kept under the high temperature. What happened? The bond formation is break. As a result, the double standard DNA molecule split into gives a 2 single standard molecules. After that, we are introducing the which one prop, the exact copy of prop. Okay, so and then the TNPS as well as the polymerase reactions also carry out. Polymerase enzymes also act upon it. As a result, you will get the copy. So this is the first step. This one is also going to produce the exact copy of the DNA molecule like this parental. Similar to this one, the other part means the complementary gene of this one. Okay, so complementary gene of this one. Okay, it's also produce another copy. So this is 2. Again, they will also enter into the another things reaction. So likewise, this is the second cycle. This is the first cycle. You will obtain the 2 copies. Again, the 2 copies are split means you will get the 4 copies. 4 and 8, 16. 16 to like that. Its copy is also multiplied. It is also denoted as a n power 2 power n. Okay. The polymerase chain reactions is very, very important. The number of copy within short period, how many copies they may be obtained? 2,000 copies may be obtained. Already we studied in the previous session. Within short period. So these are the important, my dear. So don't forget, this is very, very important. See that one. The DNA molecules also splitting takes place. That is called as denaturation with the help of the high temperature. After that formation, what happens? You have to introduce the prop. Then only the leading process and also taking place both aside the DNA combination. Complementary genes also formation takes place. After that, what happened? It entered into the second cycle. Same process also repeated again and again. Okay. Finally, what happened? The number of copies will be increases. Here, how much is the 2 copies? Here, 4 copies. Again, 4 into 2, 8 copies. 8 into 2, 16 copies. That's why they denoted as a 2 power n. Finally, what happened? Number of copies. How many copies we need? Thousands or millions. Within short period, we will obtain. After that, we will finish it the polymerase reactions with the help of stop codon. Okay. So this is very, very important, my dear students. We will finish it up to this level only, my dear students. We will see the remaining things on the next classes. We summarize that. Today, what we are going to discuss, the annealing is one of the process. Okay. How it will be? Annealing is nothing but the heating process. That things already we discussed now only. Okay. During the polymerase chain reaction, what we are going to give, how it will be separated in the 2 strand means when you are giving the heat, what happen automatically it will be split. Bacteriophage means it's nothing but the virus that infect the bacteria is called as bacteriophage. Capsid means what is it? Is nothing but the external protein coated of the virus particles is called as capsid. Okay. Inside capsid, there are so many capsomeres also there. Okay, particularly tobacco mosaic virus, it contains 2,384 capsids also there. Cloning is nothing but one of the process producing many identical copies of the organism and cell. Examples we may call as Dolly. Okay, so Dolly in the year 1996. Sorry, 1996. Okay, it's done by Ian Wilmot. Dolly is one of the goat. Okay, so goat. Don't forget. Okay, Ian Wilmot. Ian Wilmot. What is the name of the scientist? Ian Wilmot. Okay, this is very, very important, my dear students. Cloning is one of the method. Okay, this is also very, very important. And the next one is already in our cloning vectors. Cloning vectors is nothing but the small self-replicating DNA molecule into which foreign DNA is inserted in the process of the cloning genes or other DNA sequences, plasmid, bacteriophage, and also cosmid are any examples. This is the structure of the plasmids. Okay, next one is already in our complementary DNA. How it will be? RNA2. Which RNA? This one is called as mRNA2. mRNA is nothing but the messenger RNA2 DNA with the help of the enzyme is called as transcriptase. This is called as which one? cDNA. So this is called as a cDNA. Okay, cDNA. Okay, with the help of the enzyme transcriptase. Downstream is also one of the process, separation and purification of the product from the fermentation process. Downstream. Okay, step-by-step processes. Endonucleases, it's nothing but one of the enzyme catalyze the cleavage of the DNA molecule. internal position, cutting DNA at a specific site that is called as polyendonic site. Exonuclease is complete removal of the nucleotide from the any one of the end of the double-stranded DNA molecules. That thing's also already saw it. And then this is the cut. Already we saw that diagram also and deeply. And the exonuclease and the endonucleases, how it will be acted. This is the exam locations of the splitting molecules that act upon the DNA molecule that is called HIND3 and also E. coli 1. Okay, here exonucleases 1, polymerase exonucleases 1. So what are the differences between these two? Already we know about what are the example has given. E. coli is a universal enzyme. Don't forget, my dear student, universal enzyme. At the same time, carboxypeptidase is also one of the enzymes. Okay, so gel electrophoresis, it is also very, very important for the separation of the molecule, particularly in the form of fragments. Okay, that thing is also very, very important. Ligases, after the cutting of species, okay, we may join together with the help of the enzyme is called as ligases, or is called as DNA ligases. Micro injection is nothing but one of the instrument and also technique used for the DNA or RNA in the nucleus of the protoplasm of the cell. With the help of the micropipette, we can measure the micropipette 0.2 microliter, 0.1 microliter, and likewise we can isolate or taken from the samples and also inject into the target cells. Okay, it is made up of gold. It is made up of that instrument is made up of which one, you know, one of the metal is called as gold one. Origin of replication simply called as ore. It is a sequence from forms which replicating starts where there is a replication process starting. That place or that origin is called as origin or origin of the replications, simply called as ore. And any species of the DNA when linked to this sequence can be made to replicate within the host cell. Okay, so this is electrophoresis, already we saw it. Polyendromic already we discussed. Plasmids already we discussed. Extra chromosomal body is present in the circular form of any one of the plant or animals. Okay, polymerase chain reaction. What will be the polymerase chain reaction? What will be the uses of the polymerase chain reaction? We will see the remaining things on the next class, my dear. Thank you so much for the participation. Thank you, my dear. Vijay.
Thank you, sir.
Thank you, Gana. Bye. Take care. Happy Christmas. Bye.
Thank you, sir. Same to you, sir. Bye, sir.
Thank you. Thank you, Gana. Thank you.